This is Lluis Montoliu from the National Center for Biotechnology, and I'm going to be sharing a presentation on genetic edition and retinal pathologies. As you know, these are the tools that have been discovered by many different researchers, including Francis Mojica in Alicante, and for which Emmanuel Charpentier and Jennifer Doudna got the last Nobel Prize in Chemistry for proposing the use of these tools for cutting the DNA and thereby being able to edit the corresponding gene. This is done because upon this double-strand break, there are two ways of repairing this, and one way is on the left. This is the non-homologous end joining, in which several insertions and deletions are introduced in order to restore the physical continuity. And normally this ends up in the gene disruption. Whereas on the right, you have the homology-driven repair system, in which a template is used to drive the incorporation of several nucleotides. And this is the so-called gene edition. There are many different applications in which you can apply these genome editing tools in any gene, in any animal, such as for deletion, insertion, replacement, modification, labeling, etc. Disrupting is the easiest of the technologies, and deletions is also very easy and reproduce some of the genetic alteration that are found with congenital diseases. We have been applying these CRISPR techniques to investigate albinism. Albinism is a rare genetic condition affecting 1 in 17,000 people. It's characterized by a reduced vision. As you know, they have reduced visual acuity. People with albinism, they're visually impaired. They have foveal hypoplasia, abnormal chiasmatic connections, and etc. Genetically speaking, there are more than 22 genes that are associated with mutations that are causing a similar number of types of albinism. This is probably the commonest type of albinism, with mutations in tyrosinase. This is the oculocutaneous albinism type 1 that we have been investigating using the corresponding mouse models. We also do genetic diagnosis in the lab with the CIBERER of rare diseases, and similar to what happens with other rare congenital diseases, we fail to determine the genetic cause of at least 30–40% of our patients. One of the reasons could be that the mutations might be lying in the so-called non-coding genome, which represents the majority of our genome, the 98% of our genome, whereas the 20,000 genes that we normally are investigating are only occupying about 2% of the genome. This is true, for instance, for the tyrosinase, and this is what we have been investigating using the mouse tyrosinase locus with these color balls representing regulatory elements, in which we have been using CRISPR-Cas left and right to promote the deletion of this regulatory element and to study the resulting phenotype. And as you can see, upon removal with CRISPR-Cas of this regulatory element, we have an obvious drop in pigmentation, which is a robust one because it happens in different lines, and we can do genetic analysis. This is demonstrating the relevance of these regulatory elements in the tyrosinase gene. Thereby, these elements can also be important for the genetic diagnosis. We can do also inversions or duplications, or we can do point mutations. And this is something we have been exploring with the so-called avatar CRISPR mice, in which we are using this metaphor from the science fiction movie, because we are transplanting into the mouse models the mutations that we are recording in human patients. This is a person with albinism, has oculocutaneous albinism type 4. She's missing the letter C, a cytosine, in the SLC45A2 gene, and exactly the same mutation has been transferred to the corresponding mouse model. And this is a mouse that we can use for validating treatments, safety and efficacy of these treatments. In the case of the albinism, for instance, using DOPA or the proposed drug nitisinone. We can also do knock-ins, and we can also inactivate the nucleases by just turning into an activation, specific activation of a gene with the dead Cas9 and an activating domain, or a repressor domain for inactivating the gene. This is the so-called switching on and off that has been recently described, that is associated with some of the recent applications. Not everything is positive in CRISPR, and we can have the risk of inactivating other genes that are similar. These are the so-called off-target effects, which can be reduced to the minimum. But also we have the on-target effects, the mosaicism, the genetic mosaicism, because the repairing not only is produced in one single molecule, but in every different molecule, they have different ways of restoring the sequence. This is normally seen by different sequences, among which we will be finding the one we are interested in. This is a mouse that should be uniformly pigmented, but it is not, because only a few cells have been correctly restored. And this is something that we can deal with in mice by simply breeding, crossbreeding, and then selecting in the next generation those animals that have inherited only the allele, only the mutation we are interested. Of course, this is something we cannot do with human patients. How to overcome this off-targets and this mosaicism effect with base editing. In the base editing, we're using a deaminase. This is proposed by David Liu to change the C with a T or an A with a G. And this is something that has been already explored with the progeria model in mice, in which they have changed the C with a T so they can restore this disease, and the mice, they can live double amount of time. This is also been tested in non-human primates for reducing the amount of cholesterol in the blood. Prime editing, it's also been recognized as an opportunity for therapeutic, in which reverse transcriptase is used to direct the construction of the complementary DNA strand. If we compare prime editing with the standard gene editing, we will see that prime editing has a similar correct on targeting, but has a much lower mosaicism, which is relevant. This is the future, especially for gene therapy, the CRISPR-Cas technologies, which mainly means we are hoping to fix a mutation and get the gene corrected, either because we are substituting a small part of the gene or the entire gene, or because we are adding a gene somewhere else in the genome, and that was traditionally done with lentiviruses. Nowadays, we use these adeno-associated viruses, in which we can add the new genes as an episome without affecting the genome. And this is, for instance, what has been done with Luxturna, that is being used for treating retinal degenerative disease, recently approved also in Spain, with a copy of the RPE65 gene that is inserted intraocularly, subretinally by surgical means. There are many other genes that have been already approved, here listed in blue, for retinal degenerations, for let's say, for achromatopsia, for glaucoma, and other pathologies. In reality, what is happening today is that we are using CRISPR tools for inactivating a gene. And through the inactivation of a gene, we are creating this therapeutic opportunity. There are currently 46 clinical trials that today are using CRISPR in human patients, and most of them are ex vivo. Ex vivo meaning that we are obtaining cells from the patients, editing in the lab, and then returning to the patient. But this will not operate for eye-related disease, for retinal disease, in which they have to be used in vivo. And this is probably one of the few in which CRISPR is being used. This is for treating Leber congenital amaurosis type 10, restoring a mutation of the CEP290 gene. In this case, this is using two different CRISPR-Cas complexes to remove an aberrant point mutation and an aberrant cryptic signal in one of the introns that is leading to a disruptive expression of that particular gene. In general, this is being used applying adeno-associated viruses, in which we can deliver the CRISPR to all the relevant cells, but also nanotechnologies and nanoparticles are being used. So let me finish by just thanking my group and the different institutions that are providing us funds for the experiments.